So after a wonderful talk by Peju, I also want to continue on the topic of viruses, specifically nidoviruses, and focus on a step of their replication, the infection cycle, which is the replication, which they carry out in specialized compartments in the cells, very intriguing compartments. And we in Leiden have been studying these replication organelles for many years using classic EM approaches, conventional samples, so more from the cell ultrastructural side, and more recently with cellular cryotomography. And the story I will tell you today, I think, highlights the potential of cellular cryotomography to do cryotomography in situ, not only to study, as has been emphasized before, known macromolecular complex in their natural context, their spatial distribution, but also to discover new macromolecular players. So you may think that you don't know anything about nidoviruses, but you will realize very soon that you are wrong when I tell you that coronaviruses is one of the families in this order. So coronaviruses are, nidoviruses are envelope virus with positive sense single-stranded RNA genome. The order has grown enormously in the last years thanks to the advances in viral metagenomics, and currently comprises 14 virus families and more than 100 virus species, which infect an amazing range of hosts, from humans and other mammals to insects and reptiles. So I will talk today about coronaviruses. But I'm going to also talk about another family, arteriviruses. And arteriviruses are the ones that we know so far do not infect humans, but they infect a variety of mammals, and some are very important for the livestock industry. Another interesting reason to study arteriviruses, and that's what we have been doing over the years, is that as you can see in this phylogenetic tree, they are very distantly related to corona. So the features that we find in common, the similarities, likely reflect conserved themes across the order. So all nidoviruses, despite the difference in the size of the genomes, share a very similar genome organization. Coronaviruses have some of the largest RNA genomes, 30 kilobases, and arteriviruses is the smallest in the order, with 12 kilobases. But as you can see, despite these differences, the organization is very, very similar, and they also share similarities in the replication cycle, which I will illustrate a bit with coronaviruses, although it has been mentioned before, but a quick recap. So after binding and entering the cell, they release their genome, and this mimics messenger RNA, so it can be translated by the host ribosomes. But from the genome RNA, only the first 2/3 of the genome is translated into two large polyproteins, PP1A and PP1AB, and as we heard yesterday, this is controlled by a ribosomal frameshift. And these contain proteolytic activity, so the large polyproteins are cleaved into different subunits, 16 for coronaviruses, which are called non-structural proteins. And collectively, the non-structural proteins really ensure that replication takes place in a variety of ways, but interfering with the immune system of the cell by, well, we'll see later, manipulating cell membranes, and of course they also contain enzymatic activities that are required to copy the genome, the RNA-dependent RNA polymerase. So once they are expressed, replication can proceed, and this happens via a -strand RNA template. But to express the last part of the, is this working? I don't think so. Well, anyway, my pointer is not working. No. Okay. To express the last part of the genome, nidoviruses also synthesize a nested set of subgenomic RNAs that will give rise to the structural and so-called accessory proteins. And this strategy, this nested set of, okay, I'll try. Okay, that's good. Yeah, that's what gives names to the order, because nest is nido in Latin. Okay, so the structural proteins, of course, assemble into new virions, which are eventually released from the cell, and a new cycle of infection can start. So, but one of the most striking things that one sees when one looks at nidovirus infected cells is the drastic remodeling of the cytoplasm. So in this example is a cell infected with SARS-CoV-2. You can see in the periphery, the other one, the viral particles that are being released. But if you look inside, what is really striking is all this proliferation of anomalous structures that one does not see in non-infected cells, which have been produced only 8 hours after infection. And these structures, as I will show you later, are related to one particular step of the infection cycle, which is the replication, the process of viral RNA synthesis for genomic and subgenomic RNAs. And therefore, we call them replication organelles generically. The formation of replication organelles is not exclusive for coronavirus, actually, or nidoviruses for that matter. Actually, all positive strand RNA viruses that infect eukaryotes seem to use the same strategy, and this is a very important group that includes very well-known and emerging pathogens. So this is a common strategy, associate viral RNA synthesis with modified intracellular membranes, and this, of course, this compartmentalization could have a number of advantages. So it will help concentrate relevant factors, increase efficiency, but we also think that it's very important that they serve as shields from innate immune sensing, in particular during the viral RNA synthesis, double-stranded RNA intermediates are formed, which are a very strong trigger of the innate immune response. So different viruses hijack different intracellular membranes, and there's like a variety of morphologies. So let's look a bit about what the nidovirus replication organelles look like. And well, they're actually a bit more complex. They form many different structures, but the most abundant and common for all nidoviruses studied so far are these double membrane vesicles, or DMVs. I will use this abbreviation a lot, that already Peyu mentioned. So there are hundreds per infected cell. They are derived from the endoplasmic reticulum, with which they have connections, and they also interconnect through the outer membrane, forming together like a large reticulovesicular network. And this is common for corona, arteriviruses that we study, and recently it has been also shown for toroviruses by the group of Dolores Rodriguez. Interestingly, and this will come later, in this type of preparations, which are resin-embedded samples, the inner membrane seems to define a closed compartment, and so with no openings to the cytosol. So we know now that these DMVs are the central structure, the central hubs in viral RNA synthesis. After some controversy in the field, newly made viral RNA localizes in these structures. And another very interesting observation is that double-stranded RNA, which, as I told you, is an intermediate that is formed during infection, accumulates inside these structures, in the lumen of these DMVs. So collectively, this suggests that RNA synthesis is taking place inside the DMVs. And this, for us, makes a lot of sense, because we believe that these DMVs provide the kind of secluded compartment that we think would be advantageous for viral replication. But there's a problem, because the synthesis of RNA includes genomic RNA and subgenomic RNAs that, as I told you, and both species need to be exported to the cytosol, the genomic RNA, to give rise to be packed into new virus particles, and both species to be translated and generate more viral proteins. So for a while, we really didn't understand how this system could work. We always suspected that we might be missing some piece in the puzzle because of our conventional sample the limitations of the resin-embedded stain samples. So we wanted to study these structures with cellular cryotomography. The workflow has been introduced before, but very briefly, we just grow our cells and infect them on EM grids. At some point in infection, we blast them into liquid etching. Then with feed milling, we generate this thin lamella, 100 to 200 nanometers thick, that then are thin enough to be taken to the transmission electron microscope, where we collect tilt series and can reconstruct the 3D volume. For us, it was very important to try to study these replication organelles as close as possible to the native conditions. So we tried to avoid inactivation with fixatives or other means. And then we decided to focus on a biosafe coronavirus, MHB, which is only BSL-2, so we could visualize infected cells without any treatment. So I'm going to show you now one of the tomograms that we collected, and it's, for us, was beautiful detail. So you can see these DMBs and a lot of macromolecular details like fibers that are inside these DMBs, which we know that at least for a large part have to be double-stranded RNA. The connections with the ER, well, here in the segmentation model, you can even see a virus particle that was in this area. So we saw a number of features, but I want to focus in the most striking observation, which of course was looking into the DMB membranes. We found these membrane densities that span the two membranes. And that was really a very exciting moment. We had never, ever, I want to emphasize that, seen this structure with other techniques. So we immediately thought that this could be the missing link that we were looking for. And in MHB-induced DMBs, well, there are about 10 per DMB. We also look at SARS coronavirus infected cells. In this case, of course, we had to prefix the samples inactivated before blending and processing for visualization. And I hope you can see that also these DMBs contain these complexes. And this, of course, has been also shown, confirmed independently by the group of Peter Challa, and as we just saw, by the group of Pei Yun-sang. So it seemed that this is a generic coronavirus complex. So we then applied the technique that has been also mentioned before, cystomogram averaging, and got a modest resolution for structural biology. 3 nanometers, but still this was good enough to give us some interesting information about the complex. It's a large complex, about 3 megadaltons. That's what we estimate. It's probably an overestimate because of the resolution. It had 6-fold symmetry, has a beautiful cytosolic crown that you can see here, and there is a central channel that has some constriction points, but even at the narrowest, it's about 2 to 3 nanometers, which could be sufficient to export the RNA. So that's what we think it is, it's a bioRNA export channel. But of course, yeah, it's great to discover a complex that nobody has visualized before, but this raises a number of questions. The first of which is, what is this complex made of? And interestingly, all nidoviruses have regions in the first open reading frame that encode conserved transmembrane domains. If I zoom in the corresponding part of the polyprotein for coronaviruses and arteriviruses in this case, you can see the corresponding NSPs, which is a bit confusing because for coronaviruses are NSP3, NSP4, and NSP6, and for arteriviruses are NSP2, NSP3, and NSP5, but one call it TM1, TM2, and TM3, perhaps for simplicity, and they are very different in size. Different in the number of predicted transmembrane domains, but they seem to share a common organization in the polyprotein. They flank the main protease, and TM1 contains the papain-like protease that is also used to clip the polyprotein. And these proteins, because they have, well, transmembrane domains have been the usual suspects to be involved in the formation of these double membrane vesicles. And in fact, now we know that actually TM1 and TM2 are really critical, because if we express these proteins alone in the cell, we observe the formation, and this is again resin-embedded samples, the formation of double membrane vesicles that mimic the ones that we see in infection. And just a detour, again, well, you can see that the phenotype is not exactly the same. There are other types of structures, and in a way that was useful, well, this suggests that other viral factors also play a role in the formation of these structures, but we could find intermediates where you can see the ER covered with ribosomes that is separating and curving, and this has led to a model of how these double membrane vesicles are formed. Okay, so we thought that these transmembrane proteins were also likely candidates to be part of the core, and in particular NSP3, the first one, which is very large for coronaviruses. And to investigate this hypothesis, we use a recombinant virus that has GFP fused to the N-terminal domain of NSP3. And this virus was developed in the lab of Mark Denison to follow infection by fluorescence. And for us, the fluorescence was not so important as the extra 30, almost 30 kilodaltons that this fusion tag will add to NSP3. So we repeated the same workflow. These DMBs also contain force. We boxed them out, did tomogram averaging, and when comparing the new structure with the wild type, it becomes apparent that there are some extra masses in the prongs of the cytosolic crown, which are more obvious in the difference map. And this was very important because it tells us that first confirms that NSP3, composite copies of NSP3 are composed in the core, but it also gives us a hint about their orientation with the cytosolic part facing the prongs with the N-terminal domain in the prongs of this crown. I must say that these 6 copies of NSP3 only amount for about 50% of the total estimated mass, so there must be other viral components in the core, and we don't know what they are yet. A really beautiful feature of doing cellular cryotomography is that you can capture the complex in action and in different states. And it's very interesting to look at individual pore reconstructions. And when doing that, what became very apparent is that there are like variable mass densities that seem to associate both on the side, on the luminal side of the DMBs and in the cytosolic side. In the subtomogram average, this shows us somewhat blurred densities in the bottom and in the top. And again, we can only speculate at the time that an attractive possibility is that the luminal densities could be part of the replication machinery, where the cytosolic densities could interact with the nucleocapsid protein, the one that packages the genomic RNA into ribonucleoprotein complexes. And this was also, this hypothesis also supported by the fact that sometimes we saw densities that really look like the RNP, but it's difficult to tell. And also because we know that the M protein specifically interacts with the N-terminal domain of NSP3, which we know now is in these cytosolic crowns. So basically, the idea or the model that we put forward at the time is that the DMB pores could be coupling viral RNA synthesis, the export with encapsulation. Another question that we had at the time, of course, having worked so long in, also with arteriviruses, is whether this pore complex will be also in the DMBs induced by arteriviruses or other nidoviruses. So to this end, we studied cells infected with 2 arteriviruses, porcine reproductive and respiratory syndrome virus, PRRS, and equine arthritis virus. And here you can see a tomogram of a cell infected with PRRS, where you can see some budded virus particles, DMBs containing this fibrillar content. And I guess what I want to point out here is that we also have this type of structures which seem to arise from 2 DMBs that have merged. And these structures also are present in coronavirus infected cells, although in these preparations were particularly common. So, coming to the question, are there this type of densities in the DMBs of the induced by arteriviruses? Yeah, the answer you may have even noticed before is yes. So we found very abundantly this type of transmembrane densities. That you can see here in side view and cross sections from some of these structures that were on the top or bottom of the DMBs. And the complex from these images appears to be a bit smaller. It does not have the beautiful cytosolic crown that we saw for coronaviruses. And I must say we are still struggling trying to get do subtomogram averaging, and it's a bit unclear. We haven't used yet all the tricks in the book, so we are busy with that. But we don't know if it's because it's smaller, it doesn't have the crown which helps with the alignment, or because it has more intrinsic variability. In some views, however, we do see hints of a 6-fold symmetry, which suggests a similar global arrangement as in the coronavirus DMB pore. A very, very interesting observation for us was to see that about 20% of the pores seem to be associated with these structures, which are very distinct and easily recognizable as nucleocapsids, which we also see in other regions of our tomograms, budding into membranes of the secretory pathway or already budded inside these compartments with the dimensions and features that fit previous descriptions of these virus particles. And there are some even striking instances where you can see a fiber that seems to go towards the pore and then tether the nucleocapsid. So this supports the hypothesis that we have for coronaviruses, that perhaps this pores helps couple the export with the encapsulation. Okay, so an interesting system that we had for arteriviruses were cells that express, it's not infection, they just express fragments of the polyprotein. So we decided to look into that. So in this case, with cells expressing a fragment that includes the 3 transmembrane domains, and they contain also the protease activity, so these are self-cleaving polyproteins. And when we look at cells, we could find DMBs that resemble very much what we saw in infection, with the connections to the R, all the typical features, and also they were really containing these transmembrane densities that at this resolution seem similar to what we find in infection. So these tell us that even though the pore may be interacting with other elements, in the absence of the M protein, RNA replication, or the replication machinery, a basic pore structure can be formed. We could even shorten this system using another construct that only contains the first 2 transmembrane domain proteins. And in this case, you can see that the DMBs are really a bit different. They are larger, more irregular. Cryo-EM emphasizes these differences, and very often open, which really suggests that the 3rd transmembrane protein is required for a proper DMB formation. But nevertheless, these double membrane vesicles or precursors contain also these transmembrane densities, again very similar, and actually in high numbers. So it seems that the 3rd transmembrane protein would be required somehow in the process as well. And again, so it seems that only the first 2 transmembrane domains would be proteins would be enough to form a basic pore structure that expands these 2 membranes. So this is my summary. So what I have shown you is that taking together all these studies show that double membrane spanning pores are a common feature in the double membrane vesicles induced by very distantly related coronaviruses. So perhaps they are also shared, suggests that it will be also a feature for across the other. Well, these conservations suggest that they have a key function in the viral replication cycle that we believe is this viral RNA export. The data suggests that they are central hubs that couple viral RNA synthesis and encapsulation, and I'm afraid that we have many questions still about how they are formed. We know that if we take everything together, that the first 2 transmembrane proteins are sufficient to form a basic pore structure. Of course, without high resolution structures, we don't know if those pores are identical to the ones that are formed, for example, in infection. And we hope that the cytogram averaging will help us answer these questions. And it seems that the transmembrane containing proteins are likely components of the pore, and we believe that the first transmembrane domain, as we saw for coronaviruses, will be forming the pore. And of course, there's a lot more to learn. That's the problem when you find something completely new and a bit intractable, but these viral pores are a very interesting and novel class of complexes, and may, well, in the future, who knows, an interesting prospect is that they could be target for antiviral interventions. And with that, I just want to thank the people involved in this work. This has been a work over many years. Mainly, the core team is at LGMC in Leiden, where we work, and it's my group. And especially, I have to highlight the role of Pierre Wolf, who has collected and processed all this beautiful cryo-EM data. A very talented PhD student who is about to finish and move to the EMBL. We are embedded in the EM section. I want to thank Bram Koster and all other team members there for making a wonderful environment, and Eric Schneider, who is the hardcore nidovirologist. Our international collaborators, Nissan, who has been instrumental, the national facility, also belonging to Instruct, that has been instrumental to collect the data, and the funding agencies, and of course, all of you for your attention.